Hi there, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest in the United States of America. Today is the 13th day of June 2022. Here we're going to enjoy a general membrane lipid biochemistry lecture. I'm giving you this particular lecture because we're making the processive change from discussing cytoepigenetics to a new topic. And that new topic is going to include how membrane fluid dynamics relates to biochemical changes, both intracellularly, which we've been concentrating on almost exclusively for 20 lectures, and the extracellular milieu, particularly the immune environment. So that's going to be the new arc of lectures. It's going to be a little bit intermittent over the next week or two of both video and audio because I'm trying to get a lot of writing done. And when I'm writing, it is a useful thing to be able to do lectures because they co-stimulate. But it depends on how much time I have to really procure for that writing because I'm under some pretty tight deadlines. So you'll excuse me if I don't do these regularly. I will switch back to audio when I'm talking about research papers, as long as I don't want to show you data. If I want to show you data, we'll go back to the video. Video takes more prep time and more production time. And so because of that, I don't do as many video lectures as I'd like to do. But I think that we're getting a good, having two almost in a row now. I think we're doing pretty good because we've been doing a lot of audio lectures. I know you're aware of that. So let me hide this little bar here and get right into it if I can. So let's go to, see your slideshow, and from the current slide. There we are. All right. So enough of all the introductions. I'm sure you know who I am by now. So we know that membranes form specialized compartments. We've been spending a lot of time talking about that. There's such things as the nucleus, the mitochondrion, the peroxisome, the endoplasmic reticulum, Golgi apparatus, lysosomes, phagosomes, endosomes, and a whole host of other not really organelles, but subcellular features which act as compartments. So those are things like oil bodies or protein bodies. And of course, the entire system that's associated with invasion of either infectious agents or the incorporation of proteins and lipids for digestion. And these would be autophagolysosomes. And these are not the same as endocytic vesicles, which will transport extracellularly, or at least extracytoplasmically, various proteins, nucleic acids, and lipids. No, these phagolysosomes will compartmentalize the digestive process of intracellular phenomena. Now, that's going to become really important when we talk about the immune system. And so that's why I'm introducing it now. But let's go back to this general discussion. Membranes of specialized compartments, as I just mentioned what they are, will allow you to localize specific polypeptides that act as catalysts. And those, of course, are enzymes. You also get excitation response coupling. This would be in neurons and cells which support neuronal activity, such as glial cells. And of course, energy transduction is greatly facilitated by endomembranous networks. And by this process, we mean things like electron transport chain, both in, for example, in plants in photosynthesis and in all living systems that are eukaryotic. Oxidative phosphorylation associated with electron transport chain, associated with the transduction of bioenergetics from the cytoplasm to the mitochondrion, typically. Right? So we know that internal water is compartmentalized. And yet the intracellular fluid is only about 2/3 of the total amount of aqueous that you find in a cell, the rest of it being associated, intercalated with membranes that are not just bulk fluid. Now, intracellular fluid is rich in potassium and magnesium. You also have phosphate as a major anion, and chloride sometimes is a major anion. The protein concentration in the intracellular fluid is substantially higher, which provides for an osmotic potential. The extracellular fluid is about 1/3 of all the bulk water. It's high in sodium and calcium, so there's a concentration gradient there between inside the cell and outside. Chloride is the major anion outside, but as I said, you also find it inside the cell. And of course, carbohydrates like glucose and indeed other carbon sources for fuel, such as fatty acids, usually in the form of circulating triacylglycerol associated with lipoprotein fraction, are going to be in much higher concentration extracellularly at any given moment during cellular metabolism. Of course, at times, the uptake of glucose and the uptake of fatty acids in the form of triacylglycerol or sometimes free fatty acids and immediately esterified as thioesters to coenzyme A, or sometimes reesterified to lipids, glycerolipids, sphingolipids, things we've been talking about, can occur to such a high degree that you have much higher concentrations of these organic compounds now intracellular during active metabolism or active biosynthesis as in preparation for cell cycle, right, for cell division. That box I just went over right there that showed, I'll go back to it, showed you the various compounds, ions, and substances, what their concentration is typically extracellular, intracellular. I'm not going to give you all that detail, but I put that out there just to show you there's a tremendous gradient effect between what's in and out of the cell. Okay. Now, major lipids and mammalian membranes, of course, include all classes of phospholipids. There's usually phosphoglycerol lipids. There are also sphingolipids, which are phosphorylated. And then we have glycosphingolipids, which are specific class, which are not phosphorylated. And then we have sphingolipids, which are not phosphorylated, but have, as polar head groups, the identical structures. Usually they're alcohols that you find in glycerolipids. We'll talk about those. And then the third major component of membranes is, of course, the prenolipid cholesterol. Now, cholesterol is in the membrane just as cholesterol, part of the membrane component, changing the dynamics of that membrane, essentially allowing for the incorporation of water channels in direct association with cholesterol innervation into phosphoglycerolipids and sphingolipids. But also cholesterol adds to the ordering of acyl chains because of its three-dimensional structure as a cyclopentano-phenanthrene ring. And so that ordering of fatty acids can provide for them some elongation effect. That ordering causes a bit of, in other words, membrane increase in diameter because of cholesterol content. This has been well established in both in vitro and in vivo. But you have two major classes of so-called phospholipids. You have phosphoglycerides, and those are lipids which are phosphorylated, have glycerol in their backbone. That's shown here. So you've got this 3-carbon glycerol, which makes a phosphoester to whatever that group is out there, usually an alcohol. And then you have fatty acids at the 1 and 2 position of that carbon structure, that glycerol backbone. Now, those fatty acids shown here as oxygen esters, which is normally what they are, sometimes they can be also ethers, and we'll talk about those. But those esters are labile to hydrolysis, usually because of enzymatic activity like phospholipases. As it turns out, those fatty acids are going to be really important for signaling and for the production of highly reactive compounds, such as the eicosanoids, excuse me, which are oxygenated fatty acids, usually polyunsaturated, of often omega-3 or omega-6 series. Those are positional isomers, counting from the methyl terminus, of course. So you have usually even-numbered fatty acids, because that's what comes off of de novo fatty acid synthesis in most eukaryotic systems. And you also have unbranched, either saturated or unsaturated fatty acids. And these tend to be, again, anywhere from carbon length C18 up to 20, up to 22, even 24 carbons long. So the one shown there, 20:4 delta, delta is to denote that that is a change in the saturated alkane structure. And when there's a change and you put a double bond in there, they're at specific positions. This one here, 5-11-14, is 6 carbons away from the methyl terminus and is 20 carbons long with those 4 double bonds in those exact positions. Make that, of course, arachidonic acid, which is the elongation and desaturation product of the essential dietary linoleic acid, 18:2, delta 9:12, right, which is an omega-6 fatty acid. So that's just some ideas about what you might find on those positions in that glycerol backbone. Now, the alcohol can be any number of organic compounds shown here. You can have choline, which is trimethylated ethanolamine, which comes from serine. You can have serine itself, shown there. So when you decarboxylate serine, you make ethanolamine, right? You can also have the ethanolamine shown there, and you can also have inositol. Those are just some common alcohols, and the OH group is shown for each of those, you see. So, yeah, you can phosphorylate those alcohols as well. That's what's causing further diversification of the polar head group. And anytime you think about that head group, we're calling it polar because it's an alcohol, but it doesn't have to be polar. Whenever you think about that head group, that's giving you a specificity of a molecular species of lipid, as are the fatty acid components of those 1 and 2 positions in glycerol. We're now specifically talking about phosphoglycerolipids, right? Sphingolipids will give you more elaboration. Now, you also have the sphingolipids, and that they can also be phospholipids. That's why I'm saying that a phospholipid can either be a sphingoside lipid or a glycerol lipid. So you have things like sphingomyelin shown here. Sphingomyelin is going to have a sphingosine base. It's going to have a fatty acid as an amide linkage. That's now ceramide. That moiety would be called ceramide, which is a sphingolipid in its own right. We've talked a lot about that in terms of membrane lipid rafts, in terms of induction of canonical apoptosis, right? It's 2 fundamental mechanisms for the utilization of ceramide in mammalian cells. You see that when phosphate now is on that terminus and now you have choline, this is a phospholipid. In fact, what that shows you is sphingomyelin. So you have a sphingosine backbone, then you have a fatty acid attached to the amide linkage, and that can have varying degree of unsaturation and chain length. Then you have the primary hydroxy group sphingosine esterified to that entire phosphonocholine base. Now that phosphonocholine can be delivered directly to that sphingosine hydroxyl group from phosphatidylcholine, which is a glycerol phospholipid, very common in the membrane. Okay? So you understand there is exchange between the head groups, between the two different generic classes of phospholipids, those being phosphoglycerolipids and phosphosphingolipids. Understand that. Amongst a great variety of other florid, detailed mechanisms of transport and mobility of all the other bound substituents in those monomeric lipid molecular species. And then those monomeric molecular species are going to organize not by covalent bonding to each other, but by hydrophobic interactions, which nevertheless is very strong bonding, just not covalent, that is, exchange or utilization of electrons, right? Hydrophobic interactions are going to basically build up the membrane lipid component, right? And you are also going to have some covalent bonding between membrane lipids, but you're also going to now have to add this new idea of bonding, a lot of hydrogen bonding, but also a lot of hydrophobic bonding. Right? So we have to talk about all three. Okay, and there are also ionic bridges. We can't forget those. All right, so sphingolipid, sphingomyelin in particular, is that particular structure right there. You see it. And it's prominent as the coating of certain axons to generate myelinated axons, myelinated neuronal tissue in the CNS and in the periphery. Yeah. So here are some more glycosphingolipids. You have ceramide. Okay, let me put on my glasses here because it's difficult to see with that light, which is just, of course, the X group there is just going to be the proton. Your sphingomyelin, I just showed you that phosphanolcholine. There's the structure again. You can have glucose. These would be neutral glycolipids, such as glucosyl cerebroside, which its name suggests is found in the central nervous system. You have lactosylceramide, which is a globoside. You can have di, tri, or tetrasaccharides there. So these are heavily glycosylated sphingolipids. And then you can have the gangliosides, which is showing you GM2, where you're going to have a lot of glucose, galactose, and glucosamine, right? And N-acetylglucosamine, or otherwise known as sialic acid. So cerebrosides, gangliosides derive from sphingosine as the base structure. And that differs from sphingomyelin because of the attachment to the primary hydroxyl group. And again, you can see the different ones we just went over. Then you have sterol, and primarily we're talking about cholesterol, shown there. It's almost exclusively cholesterol, built up, of course, from isopentenyl pyrophosphate and dimethylallyl pyrophosphate, C5 precursors in the cholesterol biosynthetic pathway that we went over not that long ago. And cholesterol intercalates amongst both forms of phospholipids, sphingol and glycerol. And that hydroxyl group allows for that aqueous interface, so important for water channel formation. It also gives that ordering effect shown here, right? So you have these polar head groups of, say, two-component membrane phospholipids. You call them, say, phosphatidylcholine if you like to. Then you have cholesterol interdigitated there, and that stiffens or orders these acyl chains. It prevents them from carrying on with a lot of fatty acid acyl exchange because it orders them, because it prevents that fatty acyl movement laterally within the membrane. It doesn't really necessarily decrease fluidity. It just increases ordering. So two different phenomena that do have a lot of relationship with membrane-associated mechanisms and membrane fluid dynamics and kinetics. But it doesn't necessarily mean that that ordering is a loss in fluidity. Fluidity is more to do with the ability for what's within the membrane to interact with itself between two leaflets, for example, because of the diminished amount of energy necessary for those interactions. So fluidity has to do with lowering that action potential. And the action potential, it would be a measurement of the interaction of the membrane components. And not just the membrane lipid components, but the membrane protein components as well. And with that, also all the carbohydrate, nucleic acid, and other lipid components, which may not be directly bound to the membrane. Okay? So this allows for signal transduction cascades and voltage-gated channels and ligands binding to the receptors, all of which I'm going to be getting into. Okay? So you get the idea of what's going on here. So cholesterol is most highly enriched in the plasma membrane of mammalian cells. There's less of it as you move to the interior of the organelles. There's more cholesterol in the Golgi than the ER, and more in the ER than the mitochondrial membrane or the peroxisomal membrane, but there's still some there. And there's even less cholesterol isotypically in the nuclear envelope. But sometimes there's an enrichment of cholesterol, and guess where that comes from? Movement of membrane lipid rafts, which are usually a one-to-one, but not always, of ceramide to cholesterol that allows those membrane rafts to mobilize semi-autonomously from endomembranous systems. And why is that important? Because that movement allows for the transport of cargo, and the cargo are going to be signaling molecular phenomena, sometimes induced by epigenetic alterations in gene expression. Remember all of that from the last several lectures. All right, but cholesterol is absolutely important for with huge quotation marks, this membrane lipid dynamics, both at the plasma membrane and throughout all the endomembranous system, but also because of the cargo transporting. And then one more thing I'll mention here, and we'll go into more detail later, cholesterol is the precursor to the mineralocorticoids, glucocorticoids, and so-called sex hormones. All of that metabolism occurring often within the membrane or very much closely associated with it. So, for example, the production of testosterone or estrogen. Those are products of cholesterol metabolism, right? Via enzymatic conversion to various sex hormones, but also to the mineralocorticoids and the glucocorticoids. Mineralocorticoids involved in things like salt balance, metabolism in the kidney in general, highly regulated by mineralocorticoids. And then the glucocorticoids, of course, are involved in immune suppression and are triggered via the HPA axis, the hypothalamic pituitary adrenal axis, right? Which we've talked about in great deal throughout many of our lectures. So we're just introducing cholesterol as a membrane lipid now, yet it has such absolutely integral and essential composite activities for all cellular phenomena. Cholesterol is far from being a bad lipid. There's no such thing as a bad or good anything in naturally occurring biochemical, physiological responses. So having higher or low levels of cholesterol, to borrow from general and specific theories of relativity, borrowed from Immanuel Kant's epistemology, I'll have you know, that kind of relative change in concentration of cholesterol has nothing to do with the valency of being healthy or unhealthy, which I guess could loosely be described as good or bad. What that relationship is far more complex. And we've talked about it many times in authentic biochemistry. And I'm not going to give you a generalization here. I'm just going to say it's complex. Now, membrane lipids are amphipathic. That means they amphipathic means loving both aqueous and lipid milieu. So that means aqueous would be hydrophilic, water-loving, hydrophobic, water non-loving, right? Borrowing from the Greek verbiage. So you can have this amphipathic nature of lipids, and that will provide a detergent phenomenon. You have highly hydrophobic acyl chains, and sometimes very polar membrane, lipid membrane head groups. Right? So that just in the structure of phospholipids, you're going to have an amphipathic molecular structure. Saturated fatty acids tend to be less kinked in their three-dimensional space orientation. And once you start putting in double bonds, those fatty acids, whether or not they're cis or trans double bonds, will make more three-dimensional change in direction of their potency of electrophilic or hydrophobic interaction. That's what's meant by putting kinks into fatty acyl chains put in there by double bonds. So that's why there's all this interest in double bonds in fatty acids and where they're located and what their geometry is. So why is all that important? Because that will entail a specific structure. And in biochemistry, what do we always say? Structure begets function, right? So if we have a specific structure found, and we've isolated it and analyzed it via such techniques as mass spectroscopy, for example, and we find all these different structures in membrane lipids, the next question a biochemist asks is, why are those structures there and not others? They must have some function. And the deeper we go into membrane biochemistry, the more we find very, very discrete, unique functions of specific molecular species of lipids. And we've talked a lot about this in lecture, both in open lecture, my years as a professor in university systems, and also here for the last almost 5 years now, giving you these lectures in authentic biochemistry. And we will not stop doing that. So, what are the effects of unsaturated fatty acids? As more of these so-called kinks are added, membrane becomes less tightly packed and more fluid. That means the interchange and interaction of the different acyl chains is increased. That means you're lowering the barrier energy for that interaction. Now, why is that significant? If you have membrane acyl chains moving more quickly and more rapidly, that means that whatever is associated in that membrane, such as, say, a G protein coupled receptor, is more likely to be mobilized between the two leaflets of the plasma membrane in a neuron. And if you have more of an increase in the mobility of a G protein coupled receptor in something like a neuron, what are you going to get? An enhanced amount of signal transduction cascade. So membrane fluidity is correlated very highly with biological stimulatory and destimulatory processes, right? Or so-called inhibitory processes when we're talking about neuronal action potentials. And that has a great deal to do with overall mammalian central and peripheral nervous system activities. And we talk a lot about that. It also has a great deal to do with any synaptic interactions, such as the immune synapse, something which we're going to spend a lot of time on, you'll see, in the next several lectures throughout this summer, which is coming on pretty soon. Another week we'll have summer starting, right? At least on the northern hemisphere. From the southern hemisphere, you're entering, late fall, and you're entering into the winter. So I don't want to forget the fact that there are people that listen to these lectures that are on the other side of the equator in terms of the sphere called the Earth, right? So more kinks are added, more fluidity. So in a purely aqueous solvents, you can have a micellar structure formed because of the water competing against the acyl chains because of the hydrogen bonding that occurs between water molecules and that tight electron pull from oxygen towards that hydrogen atom in each water molecule, making that water molecule polar. That polarizability of water allows for the hydrophobic effect. It's one way of looking at it. The other way of looking at it, the highly neutral non-polarizability of membrane lipids will shun water. And water is forced to then, in a micellar structure, the outer segments of a micelle. Now, what will be inside that micelle is anything trapped in that lipid structure there, shown here as an open empty space. And that could include some substances which are hydrophilic, but trapped there because of the spontaneous formation of the micelle. Remember when I was talking about coenzyme A forming micellar structures, and we talked about the critical micellar concentration to form micelles. We talked about how micelles can move around organic compounds intracellularly, not necessarily membrane associated. And we talked about how coenzyme A micelles, because of their relatively low CMC, will not allow CoA itself to bulk move between subcellular organelles, and how that is really significant in bioenergetics. Think about CoA thioesters in the cytoplasm during fatty acid synthesis. After the thioesterase cleavage of the fatty acid from the fatty acid synthase, you often become meaning the fatty acid, often becomes immediately esterified from that oxygen ester, or even from the thioester of acyl carrier protein to the thioester of coenzyme A. Hence, fatty acids are essentially secluded or sequestered as CoA thioesters and not immediately labile to hydrolysis, knowing that free fatty acids could be potently lipotoxic to the cell, both interior and exterior. But at the same time, those membrane lipid components, like acyl-CoAs now, acyl-CoAs, long-chain acyl-CoAs, will be the substrate for glycerolipid and sphingolipid de novo biosynthesis, as well as complex lipid synthesis, transferring those fatty acids from one position to another between a glycerol backbone and sphingosine backbone, including the amide linkage. So you see how a lot of biochemistry has to do with the chemistry, right? And the chemistry has a lot to do with lipid metabolism, because lipids provide the hydrophobic milieu in which membranes can be organized and essentially generated and then degenerated during mechanisms such as autophagy or apoptosis. And that degeneration of membranes can cause a tremendous effect, not just within the cell it's occurring, but to all the cells within a tissue mass. And this indeed involves then the whole regulation of homeostasis of a living organism, written large, you see. That's why, as a lipid biochemist, I wasn't born a lipid biochemist, right? I was just born a little baby. But as I went through my baby biochemistry courses and then my graduate biochemistry courses and then my postdoctoral fellowship, I became a lipid biochemist by degrees, you see, by hydrophobicity, because I learned that lipid biochemistry was basically the major architectonic foundation of living systems. Of course, I knew that all along, even in elementary school when I first learned about cells. And how did I know that? Because as soon as cells are talked about, we talk about a limitation of what's inside and what's outside. And yes, plants have cell walls and bacteria have cell walls. But mammalian cells within the cushion of the tissue are cell wall-less. But all those cells, bacterial, plasmodial, mammalian, plant, all have membranes. And those membranes are delimited because of their lipid component. That's why they are absolutely vital to all living systems. I've had plenty of discussions, both in terms of constructive logical arguments, as well as just off the cuff conversations over lots of espresso. How I don't think that the prebiotic world was an RNA world. I think the prebiotic world was an asolipid world because asolipids don't require nucleic acids for biosynthesis, except once they're housed in cells, and proteins are the intermediate as the catalyst which form complex lipids. But hydrophobic substances, alkenes, for example, are very, very, very common in an abiotic world, whereas nucleic acid structures are not. So it's been my argument that just from a biochemical point of view, synthesis of very basic purines and pyrimidines was subsequent to the enclosure of those activities by membrane lipids. Hence, the abiotic or prebiotic world was first a lipid world, both prenyl and asolipid, I would argue. And with the incorporation of oxygen and the oxygen ethers. But before that, we can talk about thioesters or amide linkages, as I've just shown you both. Right? All right. And that wasn't an argument. That was an espresso type of conversation. Right? A real logical argument can also be generated. And I've done it. Membrane lipids form bilayers. That's one type of, if you want, secondary structure of lipids is a bilayer. But they can, of course, form those micellar structures I just showed you, or something called an inverted or hex 2 phase membranous structure, secondary structure. So I want you to start realizing that lipids, because they do have individual molecular species in association that are sequential within a membrane lipid system or even in a membrane lipid raft, that there is a sequence of individual lipids within that system. Just like there's a sequence in a polypeptide of amino acids, and there's a sequence in DNA and RNA of nucleic acids. Problem is, with polypeptides in almost all living systems, you really only have about 20 amino acids to talk about, which is plenty of permutations to give you all the variety of polypeptides that exist in nature. And nucleic acids, you really only have 5 total, 4 for DNA and then 4 for RNA, and you're only just replacing one nucleotide for another in between DNA and RNA. So we'll say a total of 5, giving you the purines and pyrimidines necessary for DNA and RNA, which are basic hereditary machinery. Consider that lipids, because they're already complex molecules, by complex means just like anything that means complex in metaphysics, complex means more than unity, more than one type of thing together at once. Complex lipids are formed because you have multiple molecular species of even the simplest of phospholipids, phosphoglycerolipids, or sphingolipids. You can have different carbohydrate linkages, different fatty acid linkages, different fatty acids in different positions, different levels of degree of unsaturation and chain length, even hydroxylation or oxygenation. That provides a much larger molecular species warehouse of individual monomeric units, all of which are nevertheless sequenated in membranes. And the sequences in membranes are not simple linear polymers like nucleic acids and polypeptides. Oh no, they exchange rapidly in real time from one leaflet to another, giving you entire macromolecular structures which can interact with one another. And many of those interactions are totally without nascent gene expression. That means the complexity of membrane dynamics is at several orders of magnitude higher than the complexity of DNA biochemical interactions and protein amino acid biochemical interactions, even when you consider all the possible permutations and changes of those nucleic acid structures and polypeptide structures. And that's what I want you to think about as we're getting close to closing today's lecture. But bilayer is one form, and bilayer is certainly significant. And you find bilayers, and you can observe something that looks like a bilayer with really sharp, even light microscopy, and certainly with electron microscopy. Right? It looks something like that, where the polar chains are interior and the apolar chains are interior and the polar head groups are external, and they form this bilayer structure. So only ends or edges of the bilayer are exposed to what could be called an unfavorable environment. But that doesn't happen because you get a closing of the membrane, right? It falls back upon itself because of that hydrophilic interaction. And I showed you that, and I could show you in some subsequent lectures how a vesicle can form. And when it does form, it closes the structure. And what is that but a protocell? Something inside, something outside, right? Once you can compartmentalize and keep what's within and what's without, you start having the ability to have communication between two different systems. The external milieu, which is usually much more dangerous to living systems, and the interior milieu, now encapsulated by the biochemistry of lipid metabolism in the form of bilayers or X2 phase or simple micellar or unilamellar structures. Because of that, you can rarefy the concentrations of ions, carbohydrates, proteins, and nucleic acids and do very specific work. And isn't that what living systems are all about? So the closed bilayer is an essential property of membrane, and it makes then for a semi-permeable barrier between the exterior. Right? You've heard all this before. Now I'm giving you more details, right? So you have lipid-soluble materials like oxygen, carbon dioxide, and nitrogen, and they will dissolve into membrane lipids as well as into aqueous solvent at various levels of concentration. Right? So CO2 and oxygen vary quite a bit in their salvation in water, and they also vary quite a bit in their salvation in lipid. So that allows for an alteration of gas exchange. And isn't that important? Absolutely, it's important in respiration, right? You inhale oxygen in the lung and you exhale CO2. The oxygen CO2 transport is carried out by hemoglobin within red blood cells that are moving through the upper vasculature in the lung, and that gas exchange is carried out at that level of respiratory mode. Now, all of that is related to the various miscibility and salvation of those inorganic gases. Right? So I want you to keep that in mind. So even though that's the case, these are a gas, and the cellular structure is either a liquid or a liquid crystalline or a gel state, right? So you have solids and you have liquids and you have gases. Straightforward chemistry. And in fact, a lot of the gas laws can be applied to an understanding of membrane fluid lipid dynamics. Okay? Even though they don't aerosolize these lipids, some of them do, right? Such as the volatile fatty acids, like acetic and butyric and propionic acids. All of those are the beginning, or that is the degradation products of either outright lipids like fatty acids or the degradation products of amino acids or organic carboxylic acids. But they're all called volatile fatty acids because of that acyl chain, right? You have that radical, that oxygen ester to the carboxylic acid, which can otherwise be, the rest of that molecule be a long chain alkane or an alkene, depending on how many double bonds there are. Right? So gases, though, can move pretty freely through a membrane, although there is a salvation chemistry to them, which we can go into depth on. Small uncharged polar molecules like ethanol also tend to be able to move through membranes relatively freely, although there are a lot of caveats to that. Things like water and things like urea require some kind of solvent mobility solution to the problem. And that can include things like generating water channels or by titrating polar groups according to the polarity of the head groups on the lipid membrane, thus carrying out a sort of ion exchange reaction within the membrane. That's right, cation or anion. Then you have large uncharged polar molecules like glucose, which almost always require some kind of facilitated diffusion, which we'll talk about in detail. And that has to do with transporters, proteins which act as transporters, like the GLUT transporters, GLUT2, GLUT4, for example. Some insulin-dependent, some non-insulin-dependent transporters, respectively. And that allows organic compounds like glucose to traverse the membrane, but selectively, according to, for example, bioenergetics, which means you're driving the metabolism of glycolysis, oxidative pentose phosphate shunt, fatty acid synthesis. At the same time, you're transferring bulk glucose from circulation into a cell. Right? You also have charged polar molecules, amino acids, nucleic acids like ATP, that would be a ribonucleic acid, and of course, phosphorylated sugars. And it's well known that phosphorylated sugars will not pass out of the membrane once they're synthesized, like glucose 6-phosphate, unless you have a phosphatase enzyme. And not all cells within organ systems have that. Hepatocytes do. But for example, myocytes don't in general, right? So because of the lack of expression of a glucose 6-phosphatase in muscle cells, all the glucose is trapped in that initial hexokinase-glucokinase reaction, depending on which cellular system you're looking at. The unusual nature of glucokinase versus hexokinase in the liver allows you to trap glucose at lower concentrations or higher concentrations, depending on the flux of glucose in the blood, being a requirement for the insulin dependency of glucose uptake in all other cells except the central nervous system and the liver, because of the compartmentalized and alteration of gene expression between glucokinase and hexokinase, exclusively in the liver. But then the phosphatase and the lack of the phosphatase expression in the muscle cells, and by and large also in neurons and glial cells. So once glucose is trapped via the glucokinase reaction, that glucose then is maintained within that cell to go through, for example, glycolysis, or sometimes glycolysis, the oxidative pentose phosphate shunt as well, right? For example, in red blood cells. So I'm going to stop here because there's a whole lot more I want to talk about diffusion through the membrane. And I want to also maintain my intense discussion of the lipid component. Right now, we're not talking about the lipids. We're just talking about membrane trafficking. I want to do enough of that to whet your appetite, because it's aqueous, right? But then I also want to be able to move right back into lipids and how lipids are playing such a significant role, even in these relatively common transport mechanisms. Okay? So I'm going to stop there. And to do that, I've got to close this show. So I'm making these video lectures slightly shorter than I usually do. Normally they'll go over an hour. I'm doing these now hopefully under an hour. And I'm doing that because I want to be able to produce more of them. And as I said, production of the video lectures takes more overall work to do. But also because I want to make sure that I bring in more brilliant minds to listen to these video lectures. I noticed that there are a far larger audience for the audio lectures than they are the video, even though the video gives you the images, which allows me to point to things like I would in open lecture. And also you get to see the professor talking, which some people find amusing. And the audio, of course, you lack that. exposure. Right? But okay, so I'm going to stop here with that little bit of reasoning why I'm making these video lectures a little bit shorter, and also hopefully to keep you inspired to wait for the next one. So I'm going to stop here on the 13th of June, 2022, which happens to be birthday of my older son, and say bye for now. It's Dr. Dan Guerra.